Great. Okay. Yeah. So we'll be talking and showing examples of how you can use our software to easily sort of create video experiments with Experiment Builder with interest areas on subtitles that will eventually make analysis very easy and straightforward in terms of reading, getting reading metrics and reading measures from subtitles. Okay, so let's move on. Yeah. So essentially, we'll go through an overview of sort of how interest areas are typically generated around text, etc. And then we'll go through the steps required in order to sort of prepare your stimuli for your task. So there are a few steps. We're going to go through how, that sort of first you hard code the subtitles into the video that you want to use. From SRT files, which are sort of standard subtitle files. And then you can sort of use various tools to generate the interest area sets, which will be dynamic around the interest areas, and add them to a main experiment. So then you can run it and collect data and do your data analysis. For those who have never done this, it might seem a bit daunting because there are quite a few steps, but hopefully this presentation will make it easier. Are there sort of, just to gauge a bit, are all of you familiar with Experiment Builder and reading studies? I can see that there are quite a few who have joined, so maybe some of you are not, or but yeah, I mean, we will try to be as comprehensive as possible, but there will be some sort of basic things which we won't be able to cover here. But then we'll show you, we'll give you links to other video tutorials or webinars to get you up to date with the rest. Okay, so let's move on. So in sort of typical sort of standard reading experiments that don't involve videos, text is typically presented as a static stimulus, for instance, a page or a line of text in each trial. So that kind of task is very straightforward because our stimulus creation software Experiment Builder can very easily create interest areas around words or phrases automatically. So you can give Experiment Builder the text that you want to show in each trial, and by default tell it to use the spaces in between each word, and it will create interest areas, word-based interest areas based on those spaces. Once it finds a space, it will create the next interest area and so on. And that space is we call it sort of the delimiter character. By default, it uses the space as a way to distinguish from one interest area to the next. But you can actually specify a different character if you want to, let's say an asterisk. Code it within your text so that, and you can use that asterisk so Experiment Builder will know when to stop one interest area and start a new one. For instance, if you want to do phrase-based interest areas, or even split a word into two interest areas, you can tell Experiment Builder to hide those asterisks so they won't be shown to the participant, but instead it will use them just for interest area creation. And these interest areas obviously won't be visible to the participant, but they will be embedded within the data so that when you open the data file in Data Viewer, you can see them in Data Viewer. Okay, let's move on to the next. So these interest areas generated by Experiment Builder have two properties, which are an ID and a label. So, for instance, the ID reflects the position of the interest area within the text. So these IDs are really useful as a way to find out sort of which word the interest area surrounds. So this is an image of Data Viewer here, our data analysis software, where you can see in a trial, you can see the interest areas in this panel below. So the number is the ID 1, 2, 3, 4, 5. So here 5 is the fifth word, and the other property of the interest area is the label, which is the word that the interest area surrounds. So it will automatically adopt that word as the label. So then you can see how when you output reports from Data Viewer, it's very easy to identify which interest area a fixation is on, because you've got the ID, which tells you which word it is, and in terms of its order, and the label, which is the word itself. And then you can output, regression. You can do regression analysis and other, types of analysis that is typically done for reading, because if the participant, for instance, makes a fixation on the fourth word and then skips to the sixth word, you can tell when a word is skipped, or when there's a regression, it will be, let's say, from a higher interest area ID to a lower interest area ID. That means that the participant has saccaded back to a previous word. So these two properties are really useful. The downside of these interest areas is that they are static, which means that they are typically available throughout the entire trial, or from when the stimulus is shown up to the end. Because in such tasks, typically, there is the phrase or the paragraph of text which is there, and it's static. Okay, so let's move on. However, Data Viewer supports dynamic interest areas. So dynamic interest areas are a bit different from static ones in that you can actually specify a start and an end time within the trial when that interest area is visible and can be used. So for instance, if you have a trial where you're showing a video and you've got interest areas popping up, popping into view and disappearing, you can have an interest area or a set of interest areas that start exactly when the subtitle starts and end when the subtitle ends. Then when there's another subtitle, there will be a new set of interest area which pop up and then later disappear. So you can create a lot of interest areas. Some of them start only for the first, set of first subtitle and then disappear. Another set which starts when the second subtitle starts and then disappear, and so on for the duration of the trial, for the duration of the video. In video subtitle experiments, ideally you would have word by word or phrase by phrase or character by character interest areas around the text, so that during data analysis you can calculate reading measures for each interest area for each subtitle. So this is the problem here. How do we sort of code those interest areas so at the analysis stage we know which interest areas belong to which subtitle? So essentially, if we had to use that interest area ID and simply have the ID increase, in order, as in 1, 2, 3, 4, 5, up to, let's say, 100, if you have 100 words in the whole video, then it's not easy to distinguish. You can tell which is the first word and which is the last word, or the first interest area and last interest area, but you won't be able to tell which interest area belongs to which subtitle. So with our software, so let's move on. So it is possible to simply show a video with subtitles already burned in the video via Experiment Builder, and then play back the video in Data Viewer, draw all interest. So I mean, there is a way to, if you want to, hard code the interest areas yourself manually, draw them manually in Data Viewer. But this is very time consuming and we do not recommend it. So here we will show the steps to automate this process. So we just require the video and an SRT file. So I'm guessing you're all familiar with SRT files, but there's subtitle files, which is a text file containing subtitles and the start and end of time code for each. So using these videos and SRT files, you can automate the whole process. You can have our software automate the whole process to create the interest areas around the subtitles. Okay, let's move on. Okay, so this is how we solve the problem of identifying which interest area belongs to which subtitle. So interest areas generated by this automated process have IDs in the following format. So the first interest area will have the ID 1001. So this means that it is this first number. It means that it's from. So, well, I mean, imagine the first one is 1001, the second one is 2001, third one, except 3001. So 101. This is the first one. 2001 is the first word from subtitle 2. So 2002 is the second word from subtitle 2, etc. So the last 3 digits identify the interest area within the subtitle, and the leading digit identifies the subtitle within the video. So this is subtitle.
Let me jump in and let me say a couple of things here. Like as Kurt was saying, we want to have a way of identifying like which subtitle and which interest area which word and which subtitle each interest area goes with. We don't want to have just increasing interest area IDs because then we won't know which subtitle each one goes with. And we also don't want to have it so that the interest area IDs just start over on each new subtitle, like at 1. So we don't want to have like 1, 2, 3, 4, 5 for the first subtitle, and then 1, 2, 3, 4, 5 for the second subtitle, because when we're calculating reading measures, like we don't want words with higher IDs to have been fixated before words with lower IDs, because that will throw off some of the regression measures, some of the gaze duration, regression path duration, all these kind of reading measures that people use. So we do want always increasing interest area IDs. And we do also want to have a mechanism for identifying which subtitle each interest area belongs to. And so this one has those two properties. This system has those two properties. The interest areas are always increasing in ID value, so you're not going to run a risk of having like incorrectly flagged regressions, when you get a new subtitle that has interest areas with lower IDs than previous ones, because they're always increasing in this case. And so you'll never have a risk of falsely flagging regressions or something like that. And you have the mechanism for identifying which subtitle each one goes with. Just want to make sure that's clear.
Great.
Okay, back to you, Kurt.
Okay, let's move on. Thanks, Marcus. So this is how we're going to do it. So this is an overview of sort of all the steps that we'll be doing. So step 1, you should have a video and an SRT file. The video doesn't need to have the subtitles burned within it just yet. So just a plain video and the SRT files. Now, there are actually a lot of tools, even online ones, where you can actually feed in upload the audio of the video, and it will generate the SRT for you. It will sort of do some speech to text conversion automatically. I mean, nowadays, speech to text is quite accurate. And it will generate the SRT file for you. So I've used a couple of online ones and they were pretty accurate. So you can, once you have the video, you can actually strip the audio from the video, or if you have a separate audio file, and use that to generate the SRT file. Now we have a tool which is called Split AVI, if you need to use it, that can actually split the audio from the video, or extract the audio from the video. Marcus, I don't know if you perhaps want to briefly show how it works.
Yeah, so this is an application called Split AVI that comes
It comes with Experiment Builder, sorry.
Yeah, so when you've installed Experiment Builder, you already have this, and the way it works is you just, let me see, I have a folder here with some videos. So here are some original video files, and can add them, and then basically go to view options and specify where they're gonna be output to. Like basically you're gonna use these videos to extract the audio. And when you go to view options, you'll have this property extract audio only. And when you click OK and then click start compression, you will end up with I'll just show you in. You'll end up with these files that look like I've already moved them into a folder, but this would be the output of the Split AVI. Is just these WAV files that are just the audio, and you could use those to generate the subtitles if you don't already have SRT files. Okay.
Yeah. So the idea is that sort of the audio file from the video is enough for some software out there to generate the SRT files for you. You don't need to upload the entire video, which could be very big.
Can I jump in here? Just to say that we're all subtitling people, so that doesn't really work with the audio, but that's okay. Could we explain,
I see. Okay.
Could we explain to everyone why we're splitting the audio and video? Because I don't think people understand that we're not really playing the video, but what we're doing is we're showing them a slideshow of every single frame, kind of, to simplify things, if I'm correct me if I'm wrong, right? So we split the video into single frames and we play the audio in parallel. That's why we're doing this, right?
Well, so essentially, when you split the audio, you can do two things with the audio file. You can generate an SRT file if you don't have one, just from the audio file. Now, an SRT file is just a text file with the subtitles. So if you don't want to sort of listen to the audio and type in the subtitles and create it yourself, you can have like a tool that can create the SRT file for you by converting.
Kurt, we've got professional subtitling software to create subtitles, so we're good here.
Okay,
I just wanted everyone to understand why we're splitting the video and audio.
You're right. So the other reason is because you'll need it in Experiment Builder, because Experiment Builder, when you present video, you need to present audio separately, as in playback. So when it playbacks the video, it also plays back the audio in sync with the video. So you can't just give it the video which contains the audio, like a normal video, and it will play it, because it will only play the video without the audio. You need to add the audio separately. So it's good that you separate the audio, or have a separate audio file.
Yeah, so we'll need those separated audio files also for the main experiment that we're ultimately working towards, just to make sure that the audio is presented. So that's the other kind of reason to use Split AVI.
And maybe we can add that, so this will also allow you, for example, if you want to play with sound, like, so we run some experiments where we took the sound away to see how people read subtitle differently. and we used split AVI there as well. And then we simply did not include the audio in some of our trials, right? So you can also do this. So you can design experiments without the audio and subtitles if you
yes.
Yeah, that's
Or, you know,
that's one of the advantages of having the audio split out separately is you can do these kinds of, you know, unusual presentation strategies that people often want to do when they're creating experiments.
you know, you could even play around with the, you know, have things play asynchronously. For example, you know, have the video play at a slightly higher speed, so the lips won't match with the audio. You can play around with those. Okay, so once you have the video and the SRT file, you can use whatever software you want, such as Camtasia, or there's subtitle editor and other tools, where you feed in the video and the SRT file, and it will burn the subtitles into the video. Because eventually that's what we will be feeding into the main experiment builder project. A video complete with subtitles and interest area files. But so interest area files are text files that experiment builder can use in order to generate interest areas into the data, so you can see it in data viewer. So in order to generate these interest area files, you would also need to use the SRT files as input into a little program that we can give you, which is an SRT to data source little program. It's really a Python script that we have turned into an app pretty much. So you can either use the app or you can use the Python script and run it via the command line. And essentially it will ask you for a file name, which will be the SRT file, and an output location, and it will spit out essentially a data source that we're going to use for Experiment Builder. Now this data source, it's like a spreadsheet in a way that we use in Experiment Builder in a dummy Experiment Builder project, which will generate the interest area files. And then we're going to take these interest area files and use them in the main project. And this is the main project that we run, you know, that the participant watches essentially. And this is how we collect our data. So then when we use this project, because we would have, this project will contain the video with burned subtitles and these interest area files. And these interest area files will generate the interest areas around the burned subtitles on the video. Now, there's an extra step at this point where, you know, sometimes because it's not always easy to match the vertical sort of alignment of the interest areas and the subtitles burnt in the video, there are ways to offset, to sort of shift those interest areas up or down so they match precisely the subtitles burnt in the video. So we'll talk about that too.
Yeah, basically, since we have these two pipelines here, one that's adding the actual subtitles to the video, and another one that's generating the interest area set files, we need to make sure that they match. And it's going to include potentially shifting the interest areas up and down by doing a little testing. And also at some point is going to include making sure you've selected fonts and font sizes correctly in these two pipelines so that they match as closely as possible when we look at them in Data Viewer.
Okay, so let's start with the first two steps. So once you have the video and SRT files, you'd need to burn or hardcode the subtitles into the video using some other application that we don't provide, like Camtasia, but I'm sure you have your own software that you're familiar with. Yeah, so typically this software would need an SRT file containing the subtitles. Okay, let's move on. Yeah, so this is, you know, Camtasia can do it, but there are other ones you can use. Marcus, I don't know if you wanted to show it in Camtasia, or shall we move on?
So we can't, let me do a quick demonstration of how you can do this in Camtasia. We played around with some other software, and it's possible in potentially in the AVI Recomp. Another one was called Video Proc. You know, there are other software packages out there, but Valentina and Agnieszka actually they were the ones who discovered this technique of burning them in, and lots of thank yous to you all for discovering that. But so let's just demonstrate what we're talking about here quickly. So let me launch Camtasia. And while it's launching, the idea is we're going to add a video file into a Camtasia project. Then we're going to add the subtitles into the Camtasia project that from the SRT file. We're going to specify the font size and font type. Sorry, Camtasia is kind of slow on this computer. We're going to specify the font size and font type, and then export a new video that's going to have the burned in or hard coded subtitles like already part of the video file. So here we go. We're not Camtasia experts, so we're just going to show you kind of like the basics of what's going on here. I have a folder here that has some original video files without subtitles. So I'll just add right here. I'll add a video to the media bin, and then I can drag it down into the timeline here. And then to add the subtitles, you can go File, Import, Captions. They're called captions in Camtasia. And I will try to find that folder. So I have a folder here with the subtitle files, one for each of those 3 videos. And you kind of have to do this one by one. But you can see when I selected that subtitle file, do you see how I added this track here? And as you move along, you can kind of see them here. And to edit the properties of it, which are going to be like font size and positioning, we can click this button. And what Valentina and Agnieszka have already done a lot of work to kind of identify some font sizes that will work when matched up with the interest area generating program that we talked about. So I'll just use those values. We found that using a font size of 60 here will work, and typically subtitles are like aligned bottom center like that. So you can choose bottom center alignment, and the generating project template that we have is set up in the same way. So we want to set that to match. And then once you've set all these things, you can go export local file and choose where you want to export it and click export. Okay. And we would just recommend stick to the MP4 video file format, because that for sure is supported by Experiment Builder. Ultimately, we need to add these videos back to our main experiment so we can present them. And MP4 is supported by Experiment Builder. I will mention, while we're kind of doing this hands-on, if you're using some other software that might generate a video file format that's not compatible with Experiment Builder, I might recommend to just go to our support forums, and under the Experiment Builder frequently asked questions, if I go here, there's a one of them somewhere in here is what video formats are supported by Experiment Builder. And you can read about
if you go to the table of contents, there's a whole section on sort of video and audio. It's sorry, in FAQ EB table of content. There.
So down here, there's
there it is.
audio
yeah.
under the table of contents.
All those are quite useful, essentially, I think.
What video formats are supported, and if you go there, you can read about the supported ones. And if you have a video file that isn't supported, you can click here to this guide on how you can use a tool called FFmpeg to convert it, or you can use some other tool to convert it. But this one, we have some FFmpeg commands that we've identified that will convert it to a known, like, friendly format. So just in case the software you use doesn't output the right format, you can do something like that. But from here, you export it, and then you'll have a new file that will be imported into your main experiment. Okay, I'm not going to export it because it takes some time, but you get the idea. Okay, so let's go back.
Okay. Yeah, so we've talked about this. Okay, so then let's move on to the next step, which is convert the SRT file to an experiment builder data source using a custom Python script which Marcus created, which is also an app now. So essentially, that same SRT file that we've used to burn the subtitles into the video, we can import it into this custom, into this little app that we've created to generate a data source to create the SRT file.
So it's this guy.
Sorry, the IAS file. Yeah, so I don't know if you wanted to show Marcus
okay, so
this step.
Yeah, let's go back to this folder here. So I have a folder just with all the different kind of things that you might need to make an experiment like this. And I've put all the subtitle files into a subfolder called subtitle files. You can see here we have the 3 SRT files. And just in case anybody's curious, I'm going to first show what it looks like. Let me close. I have too many things open here. But this is what a subtitle file looks like. You can see it's really just a text file. Each subtitle has an index here or an ID, whatever you want to call it. This is the start time. This is the stop time when it's going to disappear. And then this is the text. And you can see if the text wraps to multiple lines, the mechanism is just another line of text, and that will automatically make the wrapping. Okay, so this is what's going to be the input to that script. And what we're trying to do is convert it into something like this. I've already converted it. I'll do it again in just a second, but just to show you, this is a file. This is actually a spreadsheet. If you opened it in Excel, it would be like perfect columns or whatever. But this will go into the experiment builder generating project that will then make the interest area set files that we can use for the main experiment. And so you can see it has basically just converted that same information to a different format that is friendly with Experiment Builder, because Experiment Builder is the software that has the capability of generating those interest area set files. Okay, and so the way it works is there are kind of 2 options for running it. Kurt compiled this into an app yesterday for us so that it's a little easier to run than running a Python script, and you can double click on it. And this will be available for you to download or whatever. And it will launch. If you're opening it for the first time on Windows, it might give you a little blue window that blocks you from running it because it doesn't recognize it. And you can click like the more info button and run anyway to run it. It's already kind of accepted because I did that earlier. But if you get blocked, just tell it to run it anyways. And when the app runs, it will just bring up a little window like this. And the idea is like, we don't want to have to convert these subtitle files one by one. We want to convert them all and just make like one data source for all the subtitle files. And so we can point to an input folder. So I'll click get input folder, and it's already pointed to that folder. You can see it's the same folder I'm in, but you can point to wherever. But I would just find that folder, select it, and you can see it matches there. And then this is the output file from when I ran it before, but I'll just run it again. And this time I'll call it test maybe. And that's the name of what it's going to produce. And you click process data, and now it's made that test file, and we can open it in a text editor to see. And so you can see we have this is video 1 subtitles, video 2 subtitles, and video 3 subtitles. Okay, and so now this is ready to go into our generating project.
So essentially, it just formats the text differently, from the SRT way of, listing the subtitles. to a way that can be imported into Experiment Builder, really.
Yeah, and so it's basically providing, this is the name of the interest area set file that generating project's going to produce. Is a start time, end time, and then the text. And then if you have text that wraps to multiple lines, you can see it's turned the multiple lines into one with a \n character indicating the line break. And so you can also just edit these files directly if you wanted to, but if you want them to match those files, then that script is going to do it for you. And this is gonna be the input to the next step. So let's go back to our PowerPoint.
Yeah, like Marcus said, in case you don't want to run the app or it gets blocked by some antivirus software, you can always run the original Python script if you wanted to.
Oh yeah, that's right. So let me show you that. So to run a Python script, if you are worried about running an executable or something, like basically you can open a command prompt on Windows. This is a Windows only application. And then this is the old kind of DOS command prompt. And from here, you need to go into the directory where your script is. And so you can do this change directory command, CD, and then go in and just find the directory where you're trying to go and copy it. Like I just opened my Explorer here and then copy that path and go back here. I copied it with Control-C and then paste it with Control-V. And now I'm in that folder, you can see here. And from here to run, you have to install Python and you have to install the character det module. So let me show you how you would do that. You would do like, first install Python, go to the Python webpage and get 3.8, 3.9, some 3.x. It runs on Python 3.
Or, I mean, the most recent version would probably be best, I guess, right?
Yeah, you can use the most recent version. I use Python 3.8 on this one, but this script, because sometimes subtitles have weird character, or I shouldn't call them weird, they have unusual characters for me as an English speaker, maybe with accents or things like that aren't typical ASCII characters. And so in order to allow this script to preserve those unusual characters, we have to install a module that helps it do that basically, and it's called the cardet module. And so to install that, you would do like this. This is just the command for like running a Python installation thing, and you do pip install cardet. This is Python stuff. You don't have to worry too much about the details if you're not a Python programmer. But what this is going to do is install this module that allows us to kind of preserve those strange characters. And I've already
you might need to install pip first, I guess.
Pip now comes, I believe pip comes with Python 3 now.
Okay.
But from there, like once that's installed, it's already installed on mine, but yours would like download it and install it. From there, you could do like Python dash 3 point whatever you're using. Like if you're using 3.14 dash 3.14, and then you can I usually just type the first three letters because I can't remember the exact name. And then I press the tab key and it'll kind of autocomplete. It'll like look for files in the folder with that name and it'll autocomplete, and you can keep pressing tab to scroll through them. But you can see it found that when I hit enter, and you can see that same application comes up and you just run it that way. And it's the same thing, but that's the Python method.
Sorry, I have a quick question here now that we're talking about characters. Have you tested this with Chinese by any chance? Because some of the participants would have Chinese as their working language. And I just thought of that. Have you done any tests with Chinese characters?
That's a great question. We have not tested it with Chinese characters, but I would encourage, if anybody's going to do that Chinese character route, send us an email with your materials and we can try it with it. Mm-hmm. And if it works, great. If it doesn't work, then we will adapt it, and the version of the script that gets posted on our website. We're going to post this whole video on our website with all the materials that we're talking about here, the script, not Camtasia, but like some example subtitle files, videos, and these experiment builder projects. you can send it to us, and we'll make sure that the one that gets posted can handle that kind of stuff, or we'll do our best to make sure it will handle it.
Awesome, thanks.
Great. We have another question.
Yeah, I was just wanting to add something. So we work with Polish, which is not obviously Chinese, so it's different characters, but it has a lot of diacritics. And by installing that Cardet or Chardet, I don't know how to pronounce it, that module in Python, we were able to display the subtitles correctly in. So all of the diacritics, the on, the en, and various accents on the words are displayed absolutely fine, at least in Polish. So if you're working with language like, I don't know, Hungarian or any languages that have these diacritics, at least you are sure that it will work there.
Can I also ask Hui Huang, who's here, to send me your recent Chinese subtitling file, and I'll forward that to Marcus so they know they have a Chinese subtitling file. Thank you.
No problem. Yeah, that would be great. We can try it and then edit the script if needed. Okay, so I'll close this and.
So the app version, the compiled version, should already have the Chardet module all within it.
Great point, Kurt. So if you're using this app version, you don't need to do any of that command prompt stuff that we did just now. It's already bundled in together.
Yeah.
Okay.
Okay, and since you mentioned Chinese, with regards to the interest area creation, typically, so instead of using the default space for the interest area delimiter, sometimes it helps to use asterisks so you know exactly where to place the interest area. Where one interest area ends and another one starts. So that's something you can do while generating all this, instead of using the default space in between the words. Okay.
Yeah, let's talk about that when we get to that project. I'll show you how to do that.
Great. Okay. So SRT files have we've Marcus talked about this, covered this already. So that's we've covered this. This is the converter from SRT to data source.
This is what it looks like if you haven't installed it, by the way. This is like what the kind of stuff you'll see as it installs it. And then this is kind of
this is the little app.
Kind of running the Python version, but you can also just double click the app. Okay.
So let's move on to the next step 4, where we've used that data source generated by that little Python app and add it to a dummy experiment builder project, which will generate the interest area files. So we're using experiment builder here because it already has built-in functionality of sort of generating interest areas for data viewer. So we're taking advantage of that to generate these interest area files from an experiment builder project. So we're not going to collect data with this project on participants, but we're simply using it to generate the interest area files. Okay, so let's move on. So this is step 4 here, after the generating the data source. We're feeding that data source into an experiment builder, a dummy experiment builder project, which will give us interest area files. Interest area files have the location of each pretty much text files with the location of each interest area and its start time and end time. So these will be dynamic interest areas. Okay, which will then feed into the main project. Sorry, Marcus?
Should we demonstrate that?
The generating experiment?
Yeah.
Yes, definitely. That's a good idea.
Okay. Maybe this time.
Okay.
Let's go through these and then we'll demonstrate. I'm sorry, Kurt.
Okay. Yeah, so first we're going to open a project which is part of the package that we provide for this sort of subtitle package. We're going to open the experiment builder IAS generating project, which is called video generate subtitle IAS files. We're going to go to the data source of the experiment. A data source, it's a bit like a spreadsheet where we specify variables for the experiment. And we're going to essentially import the data source that was generated by that Python script. So in the data source, there's an import data icon that we can click on, which is here. Point it to the data source file, and click on so for the first file video,
Actually,
we can use
sorry.
overwrite.
Let's take this out. I'm sorry. We edited the script now so that it will do all the files for you at once. Don't have to do them one by one anymore. And let's just block this here, because the encoding, as we discovered yesterday with some emails to Valentina and Agnieszka, the encoding actually needs to be changed to support those unusual characters. And so we found, at least with Polish stuff, that we have to use the UTF-8. Like the output file of the Python script is a UTF-8, and so we have to import it with UTF-8, or at least the current version is set up UTF-8. If we find out it doesn't support Chinese characters, we might need to change that too. But the encoding should not be the default. Okay, I just want to point that out and edit the PowerPoint on the fly here. Okay.
Perfect. Yeah, so then we'll end up with the updated data source, experiment data source here, which will have the video file name, the onset time, the offset time, and the subtitle text for each subtitle. Now we can edit the font characteristics of the subtitles in the multi-line text resource inside the innermost sequence of the experiment. Well, inside a node called display screen subtitle action, which Marcus will show you where it is now. So essentially, we want to modify it to match the font style of the burnt subtitles on the videos. Okay. So typically we use Arial size 44 single spacing, which will match those settings that Marcus mentioned before in Camtasia. This is so that then the interest areas will exactly match will be, right over the burned subtitles. Okay. Now, here you can also edit the interest area segmentation properties, just like in any other text study. So like we said before, if you have, Chinese characters, you might want to have used an asterisk as a delimiter character. But there are various sort of word segmentation interest area preferences. If in Experiment Builder you go to Edit Preferences, Built-in Interest Area, Word Segment. So by default, you'll notice that the delimiter character is a space, and it will be shown to the participant as a space, essentially. But if you put asterisks to specify where you want an interest area to start and end, you can have it so that the asterisk is replaced by a space or disappear completely and not be shown. So these are all settings that are described in detail. We have a webinar, a psycholinguistics webinar, which talks about reading studies, how to create and analyze reading studies with Experiment Builder and Data Viewer. They're essentially the same, so you can go through that.
Let me point out where that.
Great.
So if you're just generally new to Experiment Builder and you don't have any idea how to use it, or you just want to know the basics, you might want to go to video tutorials and just go through the Experiment Builder tutorial series, which will just introduce you to how it works generally. We have a webinar. This is where this webinar is going to be posted to, or this workshop is going to be posted under this webinar section. But we currently have one implementing text reading studies. This is for static text, not subtitles, And if you just want a background on that gets a little more into details about how of text interest area segmentation considerations, then this is a good place to start. And you can see we got the PowerPoint and some example projects there. So that's what Kurt's talking about in terms of these resources. Okay.
Great.
So should we, I guess there's one more about this that.
Okay, so essentially I mentioned before, or we mentioned that sort of, it can be sometimes that the interest areas that are generated do not match precisely the burnt subtitles in along the vertical sort of orientation. So you might need to shift it vertically up or down to match precisely the subtitles. And you can do this at 2 different steps. This is one of the steps where you can actually adjust that shift. So in the experiment build-up project, in this dummy project, there is an IA position offsets variable. And Marcus will show now when we, so its value is 0, 0, which means that there's no shift. But you can add a value for the X and Y, 0 and 0 is X and Y, in order to adjust it vertically however you want. But you can first essentially run the project to, prepare the main project, run a dummy version of the task, as in collect data from a friend, simply to see whether the interest areas match. And if they don't, you can actually, there's a way to check by how many pixels the vertical alignment needs to be shifted. And then you can go back to this and change it here and regenerate those interest area sets.
Yeah,
okay, so now Marcus.
That's a very important point, Kurt. Like this position offset stuff, it is available in the generating project, but when you're building the experiment for the first time, you probably don't really want to be messing with the IA offsets in the generating project. You really can do that later in the main, as you're setting up your main experimental project, do the fine tuning, as Kurt says. This property is available in the generating project, mostly in case you want to adjust interest areas after you have already collected data. Because if you've already collected data, you can't really go back to your main experimental project and do the adjustments. It's like kind of too late at that point. You've already collected the data. So at that point, you can go back to the generating project and adjust this if needed, if they needed to go back up and down, and change the way the interest areas are segmented, like add asterisks where they need to go, and maybe move it from word to phrase level or something like that. And then at that stage, you're really just using this generating project to create the IIS files that would be added to Data Viewer so that you can have new kind of segmentation of your text. And so that's this position offsets isn't really there for setting up the initial experiment in the generating project. It's there for kind of tweaking things at the data analysis stage if you decide you need to reparse your subtitles into different types of interest areas, like phrase-based or character-based or something. Okay, but maybe I should demonstrate all this stuff now.
Yeah, that's good. So, someone's asking a question, Marcus.
Yeah, sorry, it's just me. So I just wanted to double check then. So once you import everything onto the main experiment, you then test run the main experiment. You may see that, so you should see the interest areas, and then you will see if they are out of if they're offset, if there is an offset that you need to correct, then you can go into the screenshot that you're showing here. So this is the main project, is that right? Then you can change it, and then you have to test run it again to check if this offset. So when you change those two numbers from 0 to something else, you then test run again. Is that correct?
That's correct. Well, yeah. And let's talk about that when we get to the main project, about how we can do that. That's a big part of the step. And there's usually a way to do it, like to figure it out precisely in just one try. You try it, maybe they're offset a little bit. You find out exactly how much you need to adjust them to make them correct. And then you apply that to the main project.
To the main project. So this is what we're seeing here. This isn't the main project. This is the interest area generating project. So this offset can be done if, let's say, you collect all your data and you need to regenerate the interest areas and bypass the main project and simply import those interest areas back into Data Viewer. Great.
Yeah, so that's the main reason this this offset capability is in the generating project.
Okay.
But
Do you want to go through the generating project? Perfect.
Yeah, let's do that. So I have in this same folder where I have all the materials, I have video generate subtitle IS files project. This is the dummy generating project, whatever you want to call it. I tend to call it the generating project, okay? And to launch an experiment builder, probably you can just double click the graph.ebd file and that will launch experiment builder and open that experiment. And it looks like this. It really doesn't, you don't really need to edit any of the things except potentially like this and the data source and the interest area segmentation stuff that we just talked about. So the first thing we want to do is go to the trial sequence. This is called the trial sequence, and open its data source, which you can do by clicking here or by just clicking. This is where you can always get to a experiment builder project's data source is by clicking from this drop-down menu, and you can see it. Since this sequence has a data source, it's available in the drop-down. And if I click there, it'll bring up the data source. So what we mean by data source is like this sequence is going to iterate multiple times. In this case, it's not iterating once per trial, it's iterating once per subtitle. And so this project, when you run it, it's just going to like go through it's going to present each of these subtitles very briefly on the screen. And all we're really doing is just taking advantage of Experiment Builder's ability to segment that text into interest areas and then save those interest areas set files for us, which we can then import into the main project. Okay, and so as Kurt, this one we've already imported from the output of that Python script, and so it's already populated. But let's just delete them all and act like we don't have anything there. Okay. So what we can do here is like click this import data button, and now I'm trying to find the file to import. And I'll go into the same folder. Video subtitle, subtitle files. Remember, this is where we ran our Python script. There's the executable, there's the Python script. These are the original subtitles. And this is the file we just generated. I'm going to use that one rather than the one I generated yesterday, just so we can go through the whole thing. And just click OK. And then that script, at least the current version of it, is outputting UTF-8 files. And so I'm selecting UTF-8 encoding. This may change if it turns out Chinese characters aren't supported this way. We might have to look at a different encoding or a different version of the script, but at least for Polish and English and that, this should work. And then just leave all the rest as it is. It's tab delimited and each row ends with a new line character. And so we can click import, and you can just overwrite, always overwrite, because this one table that we've just imported has all of our videos. You can see these are for video 1, these are for video 2, and these are for video 3. So this one generating project is going to generate our interest area sets, our dynamic interest area sets for each set of subtitles.
Yeah. Okay. And importantly, you don't show this to the participants. This isn't a stimulus, I mean, it's not something that you do eye tracking with. This is just a tool to generate the interest areas.
Yeah. And at that point, you can click test run. I don't know if we probably don't need to really run it. I mean, it's not very exciting. Basically, you just, I'll start it running and then I'll stop it. Okay, so it'll give you a warning. You're running in test run mode. That's fine. We don't need to deploy this experiment like we do for normal experiment builder projects. Since we're just using it in this special way, we're not really, this isn't an experiment actually.
But one thing, you'd want to check also that you've got the screen resolution which matches that of your you know, if you go to edit preferences, screen, you know, width and height, they need to match those of your screen.
Yeah.
We kind of assume that most people will be using 1920 by 1080 resolution because most monitors, you know, have that resolution by default. So that's something important. Even when creating the videos, it might be a good idea to generate the videos in that resolution too, so that they're presented at full screen, and then you don't have to worry about it.
Yeah,
It's
there are
just
several
something.
things. There are several things we want to format. Actually, we need to format the resolution here. We need to format the size of the subtitle of the text that's being used to generate the interest areas, as we talked about, and we potentially need to adjust the segmentation of which we can get to here. So I'll just show how to do a quick run first, and then we'll go back and talk about this. So
it might not show because of Zoom. But
yeah, it might not show. So I'll just say, okay, let's do it.
It might just show a gray screen.
And right now it's like building some images that aren't really used in our pipeline, but that will help it with the segmentation. And you can choose a data file name. I'm just going to use the default test. It doesn't really matter what you use. So you can stick to test in this case. And you click OK, and it'll say, in this case, I'm not connected to a host PC or iLink eye tracker, so it's going to say some eye tracking isn't. And so I've enabled dummy mode under this iLink section.
So you don't need to be connected to an eye tracker. If you want to do this at home on your own computer, as long as it's got the same screen resolution as that of your lab, you can just run it in dummy mode at home.
And you click OK, and it might disappear for you. Hopefully you can still hear me, and I'll maybe describe what's going on. So I just see a gray screen now, and now I'm seeing the subtitles appear.
Oh, it is working actually. Okay, we can see it on Zoom. Perfect. So it's just going through them quickly and generating the interest areas.
Yeah, so it'll take about, it's done now. And once it's finished, you can, this is the same project folder. Remember where I went into this project folder, video generate subtitle IAS files. I opened the project here. The interest area sets that it saves are kind of saved in an unusual location. This isn't where interest area sets are usually saved in an experiment builder project, but this is a very special experiment builder project. It puts them in the My Files folder. It's not a typical experiment. That's what I mean by it's special. It's just a generating experiment.
The My Files folder is sort of a special folder within Experiment Builder where you can put in things that are not necessarily used by the task. So it's good, you know, it's good that they're saved there because it's sort of out of the way, kind of.
Yeah, and so you can see, because I used the name test for the session, it saved it under test. And there's just a subfolder there where the interest area sets, and you can see there are 3 interest area sets, 1 for each video. And if you open it with a text editor, you can see it follows experiment builders, data viewers formatting expectations for interest area set files.
So each line is a separate interest area, essentially.
That's right. So this is an interest area. This is the time. It onsets 1.610 seconds after the video starts, and it offsets 3.610 seconds after the video starts. So these are the onset and offset times. This is the shape of the interest area. They're all rectangles in this case. This is the ID, and you can see it follows that formatting that Kurt talked about earlier, where these are all part of the first subtitle, and we have 1, 2, 3, 4, 5, 6, 7, 8. The next subtitles start at 2001 and go up to 2008. And that way they all have higher IDs. And so any fixations to previous interest areas that are no longer existing are not going to mess up our reading measures. Okay. And you can see, so these are the IDs. So we have lots of subtitles, and each set of subtitles, should have the same time. Each set of interest areas for a given subtitle should all have the same onset and offset times because they onset and offset together, right? And that's good. They're doing that. And then from there, we have the left, top, right, and bottom pixel boundaries where they were on the screen. And these values are affected by that by the IA position offsets variable that we showed you before. We'll come back to it, but they are affected by that. So if you change those values, they will affect these values. They'll shift them up or down. You almost never need to shift them left or right. It's mostly vertical, but we have that horizontal position just in case. And so you would see if you're shifting them vertically, this number and this number will be affected because this is the top and the bottom. And then you have the label finally. Okay, and so you can see these are the files that are produced. We have 3 of those files. I'll open the third one just so you can see each one has different subtitles for a different video. Okay, so that's the output of this thing. And now let's talk about these different options that you can use. That's the basics of how this works. But now let's talk about how this can be used to adjust properties of the interest areas. So first, as Kurt mentioned, we can go into, well, I guess the first thing, let's go back to the very basics as Kurt was describing. First, you want to make sure that your resolution matches the screen resolution you're going to be using on the experimental presentation, the stimulus display computer. You want to make sure it matches that because otherwise they're just going to be all over the place, not match it at all. So for sure that should match the resolution you're using on your main experiment on your display PC. Second, you can go into that, just to back up, you can go into a sequence by double clicking it. And I'm going to double click that. And I'm going to double click that to go all the way into the innermost sequence where we have this display screen subtitle action. This is the display screen action that handles the kind of dummy presentation. And you can see if you expand it, it really only has one screen resource, which is a multi-line text resource that has been added here. And you can double click on that to bring up the multi-line text resource editor, or just double click on the white space. That's another way to bring it up.
So the @ sign is just a link to the data source, essentially.
Yeah, so down here, what we have is, this is called a reference in Experiment Builder. And so we're not going to see this strange, this @ symbol on the screen. Instead, it's going to be replaced by the text that we added. It's going to be replaced by the text here. So that's why we saw the text change as it kind of went through it all. It's because it's referring to this, and as it cycles through all these different rows in the data source, it will change the text that's being presented. That kind of stuff is covered in that webinar on text presentation. But basically, this is how you can make the same experiment present different text across trials usually. In this case, we're using it to present the different subtitles. And if we open up the multi-line text resource editor and then double click on that reference, you can actually see where it's referring, like what it's referring to. And you can see it's referring to that column in the data source called subtitle text. And I mean, you can change it if you wanted to, but you just leave it at like in the experiment builder in general.
Yeah, I mean, here you don't need to change and
you don't need to
change all these things.
You don't need to change. I'm just illustrating like that to make reference to something else. You double click there and whatever. That's how this reference was made. In other words, so just double click there and it adds the reference, and then you click OK. And so now it's referring to that column, and that's what's going to make the subtitles change as it goes through them. You can also critically adjust the characteristics of the font that are being used. And this is where you want to match it as best as possible to what was created by the hard coding process using Camtasia, as we talked about earlier, that burns in the subtitles into the video. And you can like click and drag over it to select it, or press Control-A to select everything in this thing. And so I have everything selected. I just selected with Control-A. You can change the alignment here. You want to make sure, like, since we align things bottom center in the generating, we want to have them like this and like this. So you can see they're aligned, or sorry, like this, the bottom one. And so we're aligning them in the same way as we did in the Camtasia project that hard-coded the subs. And we've chosen the same font. That's critical. You know, choose the same font, Arial in this case. The font sizes are different. And that is because I used the values that Valentina and Agnieszka, they had already done the investigative work to find out that 60 font size in Camtasia matched a font size of 44 in Experiment Builder. There's going to be some guess and check work to this. One thing you could potentially do that I thought of is like you could make a still image from one of your videos. Just use some video Camtasia. You can do this. Take a snapshot of one frame of the video that has a subtitle in it. And you can add that to your projects library here, like add the image there, and then once it's added into your projects library, you can then add it as a background. I don't have any images here, but you can add it as a background. And then you can, then you can use this preview button in the multi-line text editor and try to match it as best, adjust the font until it kind of matches the still image that you've added. So you can do that kind of thing, or, temporarily change it from a reference to type in the exact same text, go back here and make sure that the text overlaps the still image that you've imported into that. And that's another way you can kind of match the font, but it's going to be a little bit of like guess and check, and trial and error, or whatever you want to call it. But essentially you can select it all, adjust the font, adjust font characteristics here, and adjust the spacing. And so we're using single 44 Arial, which gives us interest areas that match the sizes of the subtitles that were created by the Camtasia script. Okay, so that's how you can adjust the font stuff. And I mean, if you're happy with the ones with the size in the videos that we're showing here, then you can just stick to the defaults of the templates, because they're going to be set up in that same way. So that's how you change the font characteristics. And then the third thing that Kurt mentioned already is, like, by default, Experiment Builder is going to segment the text into word-based interest areas based on the space character between the words. Okay. And all the segmentation preferences are handled in the edit preferences. And if you scroll down to the very bottom and open up built-in interest area, you can see word segment. And by default, it's going to do it at the space level, at the word level, because the delimiter character is a single space. You can see that's just a space character. Let's say you decide you want to do them at the phrase level, or maybe you want to let's actually divide some words into multiple interest areas and keep everything else at the word level. So maybe we have some compound words that are like need to be divided into 2 interest areas. So let me see if I can find one here. Maybe you could consider whatever to be a compound word. So the way we could do this is take advantage of the delimiter character property in that segmentation settings. So we can go first, let's find and replace all of our space characters with star space. Oops, I accidentally just did it on that. I just undid what I did. So I'm going to do find replace, and we'll replace space with star space. And I'm doing star space because I'm going to get rid of the asterisk, but I want to keep the spaces. And then let's say we want whatever to be 2 interest areas. Then we can put a star there with no space. Okay. And maybe I don't know. I don't see a lot of other compound words. I don't see any, something maybe. Okay. Put a star there just to do a couple examples. Then go back
maybe
to
add a space, Marcus, after the last asterisk.
this one. Well, this is part of the same word, so I don't want to have
Oh,
a
you're
space.
right. You're right.
So I'm just trying to turn one
Yes.
word into two interest areas. The same technique could be, I'll show you phrase technique in just a second, but go back to preferences, and then we could do the delimiter character. Now, rather than a space, we want to do a star asterisk. And then we want to delete the delimiter. And so I check delete delimiter. And now when we run it, it will make sure that those single compound words are broken into two interest areas. We can close that and then run it, and it will be set up that way. Does that make sense? So you can use the same kind of interest area segmentation that you use in regular text studies, like non-subtitle, to change the way things are segmented here. To do one more quick example, let me try to undo. Okay, so I undid all that. Let's say we want to do it at a phrase level. The way I would do that is find and replace space with star. And so now all the spaces have become stars. And then whenever I want two or more words to be grouped together in a phrase, just delete, turn the space, like we would say promise nothing is a phrase. We delete the asterisk in between those two words, and that will become one interest area around that whole region. And then we go back to, it will become like that because we're about to set the preferences in that way. Then we go back here and choose the delimiter character is star, and enable delimiter replacement, and we're going to replace it with a space. That's a single space in there. Okay, and so now all those, in terms of what you see on the screen, you're not going to see those asterisks, but they will be used to mark the boundaries. And so wherever I put that promise nothing, you can see since it doesn't have an asterisk in there, that will be considered one interest area. So that's how you can do phrase-based segmentation. I guess one more way you could, that's kind of worth mentioning, maybe especially for languages like Chinese, maybe if you wanted each character to just be a separate interest area and not have to worry about any of this asterisk stuff. Let me just go back to the original. Let's just pretend these are all Chinese characters and there are no spaces in there at all. Then you can go to edit, preferences, and there's a feature, so you don't have to worry about this, where you can just choose enable character segment. And if that's checked, all the delimiter stuff disappears, and it will just automatically make each character be a separate interest area. So that's kind of simple, actually. And so there are different options. If you want to get in, the manual has more details about it. You can change margins. You can change how to treat white space between characters and lines, that kind of stuff. They're more advanced things, but these are the basics of it.
Regarding analysis, character-based sort of segmentation might not always give you reliable fixations on each interest area, because no eye tracker is 100% accurate. If the interest areas are going to be very small, one around each character, you might not be able to distinguish between a fixation on one interest area versus its neighboring interest area. So yeah, you need to keep that in mind.
Yeah, excellent point. And like maybe if you're gonna do something like that, just for character segmentation, you probably want to use a really large font, something like
exactly.
that. So, okay, so you get the idea. You set all this up how you want in terms of the resolution, in terms of the font characteristics of the reference in the display screen action, this one, and you set up the segmentation in edit preferences and your data source so that it's going to produce the interest areas you want. You do the test run, and then you have your interest area set files, and that's kind of the end of this step. Okay. I'll mention, I guess, since we talked about it already, there is this, this position offsets variable. When you're making the experiment, you usually don't need to mess with this at this stage because there's an there's a similar variable in like the main project and we can do it there. But if you've already collected data and you decide, oh, I want my interest areas to be segmented differently, then you can, and you like, your main project already has them set up in one way, but you're at the data viewer stage and you're working in data viewer and you decide you want to go back to phrase-based interest areas or something like that. And you want to make sure that the position is correct when you generate new interest areas, then you can use this position variable here. So the idea is if you're at the data analysis stage and you want to generate new interest areas, you can come back to this generating project that you used to make the original kind of experiment, do all those things that we just talked about, adjust the interest area segmentation properties, adjust whatever else, like the asterisks and other properties that need to be adjusted to make sure that works right. And then you can just check in your main project what position offsets was I using there and just use the same values here. And then you can generate the IAS files and import those directly into Data Viewer. So this feature here is really only intended for cases where you're coming back to this generating project at the analysis stage. But it is important to know that it is here too. Okay.
So at the analysis stage, this could essentially replace the interest areas which you would have had before, with exactly. The new ones. Yeah.
Exactly. Okay, so should we jump back in here?
Okay, so yeah,
we talked about this.
We've talked about all this. So let's go back. Let's go to the
here
to the next step. So the next step is to import the video and interest area sets into the main experiment project. So then you can collect data essentially from your participants. So this is the project that you would test run, make sure it works, and then deploy. So then you can collect data from multiple participants. So this is essentially step 5, where at this point we would have the video with blend in subtitles that we got from Camtasia, and the interest area sets, which are essentially text files with a .ias extension, which we import also into the main project together with the videos. And that's it. This is our final project here. And like Marcus said, once you collect data, even if you collect data from all your participants, and then when at the analysis stage you realize that there's an offset that you need to fix, you can go back to this, the generating experiment, recreate the interest areas however you want them with an offset, and load those into Data Viewer, which will replace the ones that were added by the main experiment. Okay, let's move on.
So should we show that?
Yes, good idea.
And
so
yeah, let's go through these first, and then I'll show with the real.
Okay. So essentially, when you open your final experiment project, which is the main experiment project, you can add the videos and also the audio files separately, and the interest area sets by going to Edit Library Manager in Experiment Builder. And the Library Manager is a way to import your any images, sounds, interest areas, videos, etc. So we're going to click on the Video tab, import the video files here that we're going to use, click on the interest area set and import those IAS files, and also on the Sound tab to import the audio files which belong to the videos. Then we're going to go to the experiment's data source, and here there are sort of two videos, so 2 trials in this case, so 2 rows. So this time it will be essentially 1 row per trial. So, if you just want to present one long video, you would only have 1 row in the data source here, but this demo shows 2 videos. So we've got the video file named the audio that we want to synchronize with the video, the WAV file, and the interest area set. And here you can also add the critical interest area list. So if you have some critical interest areas that you think might be important at the analysis stage, you can list them here, and these will be available as variables in Data Viewer. We can see this in detail when we talk about Data Viewer. Okay.
We kind of talked about this already, where
yes, so we've talked about this. So this is how we can tweak. So again, the same setting that we've talked about in terms of tweaking the offset is available in this project too. So you can do a test run, look at the data in Data Viewer quickly, see if there's an offset, and if there is, measure it, and then change it here before collecting data.
Okay, so should we, this is the critical ones that the critical interest areas that you were talking about.
Yes, so these are just so these are not going to affect your experiment. These are just the, if you need them for analysis. So when you output reports from Data Viewer, you can output also this for further analysis. I don't know if you wanted to.
Yeah, let's
go
through all that here.
Yeah.
So let's go back to our folder with all the materials. And you can see this was our generating project. This one here is the main experimental project. So this is the one you're going to use to collect data. And let's open it. So I'll double click the graph.ebd file. Try to hide this thing again. It keeps coming back. These are just the meeting controls are showing up. Okay, so this is what the main project looks like. And as Kurt mentioned, the first thing we need to do is go to Edit, Library Manager, and under the Video, Interest Area, and Sound tabs, we need to import those files. And so you can see, you can click the Add button here, and oops, I will go to wherever our folder is. Output from Camtasia. Here are our 3 videos that have the burned in subtitles already, and add them. And in this case, they already exist, so it's gonna overwrite them. And you can see a little preview here. These are just
the preview is not showing the subtitles because they start a bit further into the video, but they are actually already burnt in.
Yeah, it just shows the first frame of the video, I believe, in this preview. But it's nice here how you can see characteristics of the video. You can make sure that they match what you needed. When we ran Camtasia before, I didn't really mention this, but in Camtasia, you can set the frames per second of the video file. And that's kind of an important consideration for video. You want to make sure the frames per second can divide evenly into the refresh rate of your monitor. So like in the same section where you set the resolution, you can set the refresh rate of your monitor. And most monitors can do 60 Hertz. And so 30 Hertz is a convenient frame rate because that means since the screen can update 60 times a second, and since 30 can divide evenly into 60, in other words, every 2 screen updates could be a new frame of the video. That's going to work out nicely. If we had a video frame rate of 25 and a refresh rate on our monitor of 60, then you can see 25 doesn't divide evenly into 60. And so there'll be some cases where frames of the video might be a little later, a little earlier than they would be if you had a 50 Hertz monitor, for example. It's not like a big deal breaker or anything, but it will make it look potentially choppier or something. So it's typically a good idea to use a frame rate when creating your videos that will divide evenly with no remainder into the refresh rate of the monitor in the Experiment Builder project. Anyway, so you
also,
said that.
Sorry, could I just, before I forget, so in previous versions of Experiment Builder, you had to state what the frame rate of your video had to be, but now Experiment Builder will let you choose the default frame rate for those. So for those who are used to perhaps older versions of Experiment Builder, now you can choose, where is it?
I think it's this. Oh, that's the size.
No, yes, so it should be.
Here, apply
Yeah,
default.
apply default frame rate. So that is very handy. So it will just use the frame rate, the default frame rate of the video. So you don't have to check in advance. And
That's a
put
great.
in.
That's a great point, Kurt. And it's like it's another consideration. It's like Experiment Builder can actually play videos faster or slower than the original video file. And if you're not playing, like if this is unchecked and you had this at 60 or something, it's playing a 30 frame per second video at 60 frames per second. So it will be played like twice as fast, you know? And so it like, if you're seeing some case like that where the video is playing too fast or the video is playing too slow, it's an indicator that your frame rate probably isn't set right. So you can either check this property or you can uncheck it and make sure that this value matches. But I'm going to check it because that will ensure that the value that it's played at its original speed, and that then will be good. Okay, so back to we were talking about how you add the videos and then you need to add the sound files. And if you don't have like in Experiment Builder, as Kurt mentioned, like unfortunately, at least in this current version, it's on our developers list of things to change. But in the current version of Experiment Builder, if you just presented the video and didn't have a mechanism in your experiment to play the sound, you wouldn't hear any audio from the video. And so we needed to add those wave files. Remember that we extracted using split AVI, which I just closed. Let me just reopen it. Split AVI. Remember, we could add our videos here. We can go to options, set where it's going to output, and we can choose extract audio only. And then when we click there, it will create those audio files. And that's what you would import into Experiment Builder. And just to show you what I mean here is like, you can see this action here is going to display the video. And then because this is right after it, it will be synced in time. And so immediately after the display video, we have a play sound action so that they will be synchronized. And so we're playing the video on the screen and then we're playing the audio through the speakers kind of through separate mechanisms. Okay. And let's go back to the library manager. So we import the sound and then we import those interest area sets that were created from the generating project. And so if I go to the generating project folder, you remember they're saved in the My Files folder under the test, whatever name you use, interest area sets folder, and you can find them there. I should mention that generating project actually will also save interest area sets in the normal place in the AOI folder, but don't use those. Those are going to not have the offsets applied, and they're not going to have the times coded in the onset and offset times, because Experiment Builder just doesn't have that native capability to have the onset and offset times as subtitles. And so what that generating script is really doing is like opening those automatically generated interest area set files that will go in here and then merging them where appropriate and adding the times and doing some position offset changing so that these files that are in the My Files folder, these are the ones you should use in the My Files folder. These are going to be all consolidated, one file per video, with all the subtitle interest areas in that one. It's going to have the onset and offset times, and it's going to have the position like adjusted for them. And so those are the ones we'll add, and they're already added. So I'll just say, yeah, let's override them all. And from here, everything else is already kind of programmed. It has scripting that will handle, like this one uses some custom class. You don't really need to worry about editing that, but it basically handles ensuring that we have like markers in the data file of when each subtitle onsets and offsets. And it's set up so that we have markers of each frame of the video. It's going to be marked here. that kind of stuff. It's already set up just for video presentation. If you wanted to add questions or anything else, that would just, all the rest of that would just be added normally, probably after this node. Like you can put all your questions and stuff connected here, or any other kind of things that you want to happen in the experiment. This is just a very basic template that just presents a different video in each trial. Okay, and then when the video is over, it will just automatically end the trial. This is the mechanism for ending a trial early. You can see it. It checks, is the video completed? If that's true, then let's end the trial. And this thing will just check on repeat as long as the video is playing. And as soon as it finishes, we move on. This one also has a mechanism where you can end the trial early with a key press. And that is very useful when you're kind of testing the alignment of the interest areas to the text. Like you don't need to go through all the videos in your project to test them. You can just go through the first 10 seconds of the first video. Just wait until you get subtitles that will allow you to check it. Maybe wait for a 2-liner or a 3-liner if you have those, because those are going to be the ones you really need to pay attention to. Like the 1-liners are simpler, you know? And so maybe wait until you get to a 2 or 3-liner and then just end it. And then you can actually just press Control-C to end the whole experiment and not go through the rest of the videos. And then you'll have a data file that you can use to check the interest area alignment in Data Viewer. But you can see basically all, besides the kind of custom class marking that is marking the onset and offset times of the subtitles, stuff like that. This is really basically just like almost exactly like our video example that comes with Experiment Builder. That it doesn't have any subtitles, but it's the same kind of concepts beyond, and with the addition of the subtitle capabilities, if that makes sense. Okay, and you can see this is where we have our position offsets variable. And in this case, it looked like we needed to lower the subtitles. We've already kind of done the testing, and it looks like we need to lower the subtitles by 20 pixels so that the ones from the generating, so that the interest areas from the generating project would match the position of the subtitles that were hard-coded in Camtasia. And the idea is once you get them into here, you can do a test run just to generate a data file. If you you don't even really need to set up the eye tracker. You can kind of just make sure you're connected to the eye tracker, because it won't generate an EDF file, which we will need to do the checking in Data Viewer. You need to be connected to the eye tracker at this point so that we can run it and generate an actual test data file that we can use. But from that point, you could then open the data in Data Viewer. And just to give you a preview, like
morning promised nothing but a bullet in the brain, and yes, I longed.
Okay, so you see what I'm saying? We can see now, these are the interest areas. These kind of peach colored boxes are the interest areas, and you can click on them to see their properties over here. Notice how they're like onsetting and offsetting.
When they dumped me in the cell, I thought I'd seen a sliver of barred window near the ceiling, but now I couldn't remember. I tried counting to 1,000 to pass some time, but always got lost around 400.
He has the best voice, and it's just like so soothing. Like I wish he could read me to sleep at night. But basically, like you can see that this is the way you can test him. It's like you would do a test run of this project and then open them in Data Viewer. And that's what we're working towards, is having those interest areas in Data Viewer. Okay, do you want to go back to the PowerPoint, or should we? Yeah, let's go back to the PowerPoint.
Yeah, just a quick bit on data analysis, and then maybe we can show Data Viewer. So, yeah, so once you collect all your data, at the analysis stage, you essentially can analyze most things in the same way as you would with sort of a normal reading task, because of the way the interest area IDs were created. So do you want to move on to the next slide, Marcus? And the next. Yes. Yeah, so just as a refresher, the interest areas have a format where the last 3 numbers are the interest area ID within the subtitle. And the number leading those last three is the subtitle index. So the first number is the subtitle index within the video. Okay, let's move on. Okay, and data analysis will be relatively easy because since each new subtitle has a set of interest area with a higher ID than previous subtitles, interest areas fixations on previous subtitles will necessarily have been on interest areas with lower IDs. And because these interest areas are dynamic, the interest area sets from previous subtitles won't be shown for the current subtitles or the next ones. So, you don't have the risk of having eye movements reported in reports on interest areas which are not visible because they will simply not be there. Yeah, and so these things ensure that data viewer will not mark eye movements as false regressions, progressions in terms of reading patterns. And these things also make it so that data analysis for video subtitles is very similar to analysis for any reading study. You'll just have a lot more rows in the interest area reports that you create because there will be a lot more interest areas. So if you output an interest area report, each row will be a separate interest area. And there will be a lot more than in a sort of a normal reading study if you have a long video with a lot of subtitles. But the nice thing is that you don't actually need to create an interest period around each, bit of subtitle, or around each snippet of the video where each subtitle is shown, because of the way these interest areas, the IDs are created, where you have the ID, coded in the interest area, and also because of the fact that they are dynamic. So the ones that are not being used will disappear and will not be included in the analysis for each section. Okay, shall we maybe...
let's talk about some of these things. And I realize we haven't really talked about how to tweak the positions. So let's talk about kind of all things data viewer.
Yes, that's a good point.
So here we have just some example.
Warning, promised not.
Some example data that we collected, but let's imagine we are still at the experimental design. We haven't really collected real data, and let's imagine we had. Let me see if I can. I don't know. We had something like this. We're seeing our interest areas way too high. Okay, and we know we need to adjust the offset in the original main project, or I'm sorry, in the main project, so that they're presented at the right position. There are a couple ways you can do it. One way is to select an interest area, like maybe one of the topmost line interest areas, and take a note. So this is at 745. The pixel position of the top line of the interest area is at a height of 745. And so from there, we can select these, and then if you hold the Alt key, you can move them around. I think you can use the arrows to move them too, but basically move them to the right position, where they should be. And that looks good to me. I mean, look good enough. 745 was the original top, and the new top is 915. So that is like 100. That's extreme. It probably won't be that much of an offset, but that's an offset of 170 pixels?
Yeah, I mean, it's one minus the other pretty much.
And so then you would go back to your main project here, and we needed them to go down 170. So I would put 170 as the Y value of that position offsets. And after you change that and then run it again, the next time you open a test data file, they should be in the appropriate place, but you should double check again, make sure that worked properly and tweak it again if needed. But using that method, you should be able to get them pretty close. By the way, like -40 would move him up 40 pixels. Generally, you're probably not going to have to edit the X, but -30 would move to the left. 30 pixels, 30X would move them to the right, 30 pixels, and then you have to have an X component too. So I'll put, a Y component too. So I'll put 170. In this case, put 0. So -X goes to the left, +X goes to the right, -Y goes up, and +Y goes down. And that's how you would tweak the interest areas. So now let's go back to Data Viewer, and let's first talk a little bit about what we're just going to go briefly through like what Kurt was just talking about. So we've already got all our interest areas here. They're already appearing and disappearing as the subtitles do. And so really, and because of the ID like numbering scheme, really the rest of it is like just like any other reading study where you can go to analysis, reports, and you can use any of Data Viewer's report options for your analysis, but often people who are doing reading studies will make interest area reports, which will result in a file where each row of the output file is an interest area or different, in this case, different word. And the columns, which you can select from this dialog that appears, the columns will be like the different measures you're trying to get for that word. So for example, if you just wanted to know like the total time they spent fixating that interest area, then you can add interest area dwell time. We probably should add like interest area label and ID as columns. So we'll add those. Let's move dwell time to the right a little bit. This is the left-right ordering. The up down here determines the left-right. So this is the leftmost, and they go to the right from there. So you would have the interest area ID, the label, and then you would have the total time they spent fixating it. And if you wanted to get like how many regressions out did they make from that interest area, then you can include that. And because of our interest area numbering scheme, it's not going to falsely identify like fixations on new interest areas as being regressions from previous, on new subtitles as being regressions from previous subtitles, if that makes sense. Always increasing.
We've got a question, Marcus. I set up the Zoom call, so probably I'm the only one seeing the raised hands.
Yeah,
Sorry.
thank you, Kurt. I just thought that this maybe was a good time to ask this question, because usually if you have any additional variables that you're interested in, so you display subtitles, but sometimes you edit these subtitles to contain certain words. In our current experiment, we're interested in cognates versus non-cognates. So some only of these words, are of interest. And but it could be multi-word units. It could be, legal terms versus non-legal terms. Anything that you are interested in investigating within the subtitles, like certain words. And usually, when we are at this stage that we export the data, we will have some blue extra variables that we can export, right? To know if towards the bottom, yes, yellow in this case, great. So now, if I wanted to add information about certain keywords only in some of the many subtitles that will be displayed, if I remember correctly, we add columns to the data source. Are these columns added to the main project, or we need to go back to the subtitle generating project to mark them? Thank you.
Great question. And sorry, we meant to cover that, and it's a good reminder that we haven't covered. So definitely to the main project. Like the generating project is only really going to give you those IAS files, and the IAS files themselves are not really going to have any direct indicator of what a critical word is. And so the idea, there are different ways you could do this, but I think the easiest way to do this would be in the main project. You could do like this. And in this case, I put all the critical words into a list because these lists can vary in length. And sometimes you might have more or less critical words for a given trial or given video. And so the idea is here, like maybe this one has 403 or something. Like this one has an extra critical word. And so in this main project, we have a column that is really just used to log the interest area IDs of the critical words in the video.
And can I just ask, so if we wanted to then analyze all of the subtitles as well, right? Maybe we have certain conditions per trials, I don't know, no subtitles, subtitles in the first language, subtitle in the foreign language. And so we want sometimes to look at all of those words, including those particular legal terms or cognate words or literal versus non-literal translations, whatever. But so can we then have just all of the interest areas in each subtitle?
Yeah.
But also,
yeah, for sure. So this method, like you will still have all the interest areas in the interest area report. And this isn't going to make it so that this is not going to make it so that you only have these interest areas data in the report. Instead, it's going to make it so that you just have this additional information in the same report. So the output report from Data Viewer just always by default is going to have all interest areas from the trial. Okay. And so when you include this critical IA list variable in the output file, it will add an additional column into your interest area report that you can then utilize in your statistics program like SPSS or R, or whatever you use. And you can say, select only the cases where the interest area ID is a number in the critical list. You know what I mean? Or if you have you could have lots of these columns. If you know you have like 4 different types of critical words across all the subtitles, you could potentially just have like 4 columns here, crit word 1, crit word 2, and instead
that's
of
exactly what I wanted to ask. So if we, for example, in our current experiment that we're designing right now, we have cognates, we have some non-cognates that have been matched for word length, for frequency, for position in the subtitle, for all of this. And then we have some extra words that are not matched, non-cognates, which have potential to be learned, right? So we could have then 3 columns, or, you know,
you can have as many columns. Yeah, you can have as many columns as you want. And
then for each video, we will add which of these words, well, we will add these identifiers, right? So where the first number is the subtitle number, and then the last 3 digits are which word ID in that subtitle.
That's right. That's right. Okay. And you can have as many, you could have 10 of these columns if you wanted. I just made and they can be list types or number types. If it's list, then that same column could have a list of all like maybe this is a cognate IA list. Maybe you have 3 cognates in one trial, 2 in another, and 2 in another. Or maybe you just have 1 cognate in each trial and you just need 1 number to log it. It's really just setting yourself up for having the information that you need in the stats program to do the selection. And the interest area report is going to spit out all interest areas for your interest period that you've selected in Data Viewer. We'll talk about that in a second. And then these columns, if you choose to include them, as you noted, they're available in these kind of custom columns that will be at the bottom. We've changed the color from blue to yellow. They used to be blue and now they're yellow, but you can just add as many of those columns as you want. And then when you open your interest area report in your stats program, you can essentially compare the interest area ID to the values in this and to help you do the selection. Okay, so it's kind of up to you at that point to do the selection or whatever, but this will give you the information that you need. And I should also mention related to this, like you don't have to do it in Experiment Builder. Like this doesn't affect the participant's experience at all. It's just convenient to do it this way because then it's already there for you in Data Viewer. If you forgot to do this or you decide, oh, I have a new set of critical words that I want to do, you can either add them in your stats program or in here, like just make new variables in your stats program that are different for each video. Or in here, you can go to Analysis, Trial Variable Manager, and let's say I have a new critical word. I'll say I'll make a new variable, new critical word, cognates or whatever, you know, and you can give it a default value. I'm going to leave that blank for now because it's not useful in this case probably, but I'll say, okay, and I've added a new variable. And now if you go to Trial Variable Value Editor, you can see we have this. You can copy and paste from Excel here, but you can see I'm trying to just populate the values with the appropriate one based on the video maybe that it's using, you know, and you can just fill these in. And then that new variable, let's keep those changes, will also be available. You can see, you can include that one. So you don't have to do it at the main project stage even. You definitely don't do it at the generating project stage. You would do it at the main project stage or add them post hoc in data viewer.
Okay, perfect. Okay. Thank you very much.
So that's how you would handle the critical words. And if you're splitting, by the way, I should mention, if you're splitting these in half or whatever, remember that that's going to affect the ID. So like if they're all done at the word level, this will be like 1001, 1002, 1003. For the next subtitle, or for this one, it will be like 3001, 3002, 3003. If this is split, let me find one where it's like a good split, something or whatever. Okay, well, let's just window. I don't know, window. That's not really a split, but you can imagine if well, let's do sliver. If this were split in half, then it's going to affect the interest area IDs of it. You know, so this would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So this would have been 14 at the word level, but when we split this in the generating, it makes this, it bumps up the interest area IDs, if you know what I mean. Because now the interest area IDs aren't necessarily at the word level. For the ones that you've split in half, each one is gonna have its own ID, and they always go up by 1. Just make sure you're aware of that. Then another kind of consideration, I didn't really talk about this yet, but oops. I'm trying to move this. I have these meeting controls over there. Okay, so I've already done this in this data viewer viewing session, but like always when you're using data viewer, like the first thing you should really do after you like import all your data is set an interest period so that you're only looking at data from the relevant part of the trial. And that goes even if the only thing that's happening in the trial is a video presentation and then the trial ends, you should still set an interest period so that you're starting exactly when the video starts and ending exactly when the video ends. Because like the eye tracker recording will start a little bit before the video presentation starts, and you want to be excluding that kind of dead period before the video begins. And so the idea is like you can use the event marking messages from the main project. You know, this main project is going to send a message to mark every frame of the video, to mark the time when the sound starts, etc. And then the custom class is also going to send additional messages for each subtitle, onset and offset. And so if we go back to this data viewer thing and we look at the messages, let's select a trial here. You can see we have all these messages. Every frame of the video is marked with a message. Sometimes, since the frame rate is like 30 and the screen rate is 60, we have double because it's like, but you can see we have a message for every frame of the video. And then let me turn off the fixation so it's a little bit easier to, and there, do you see? In this case, the subtitles onset and offset at the same time, like one disappeared and the other appeared instantly. But you can see subtitle onset 4. This is the onset of the 4th title. And at that same time, the previous subtitle offset. Okay, let's see if we can find another one. And there's another one. Subtitle. So that's when the 4th one offset. And you can see if you click on it, it'll jump to that point. The point I'm trying to make is that what you're going to do probably in most analysis is first set an interest period so that you have the entire video in it. And you can do that with the interest period manager here. This is covered in our data viewer video tutorial series, but I just want to hit the highlights of it. Basically, I can call this video period. We're going to start at the message frame to be displayed 1, which is the first frame of the video. And then we're going to end at the message display blank. Like if we go back to our main project, you can see this when the video is over, we have a message display blank. And so when we set this interest period here, we are now looking at all the data for all, like we're including only the video period for all videos, basically.
Morning.
And then when you export the report, you're going to have like all subtitles for all videos in the output file. So let's just make a quick report. And it defaults to saving it in the folder of the viewing session. So I just call that IA report. And maybe I didn't. Oh, I saved it in deployed experience. I saved it kind of in the wrong place. But if I go up a level here, this is the viewing session that I just saved. And that's the report that I just output. And if we open that up, it gives a little warning because it's not a real Excel file. It's just a text file with that format. And so Excel just gives you a little warning, but that's fine. And you can see now we have, let's expand this a little bit. For each word of each subtitle, we have a row. And we have our critical list here. We have our interest area IDs here. I just included one variable, the IA dwell time variable. But the idea is like you could then, in your stats program, say, select only the cases where the interest area ID is inside the critical words. And then you would have a report that only has the critical word interest areas in it. But just by default, it's going to include all interest areas, for all subtitles, for all participants, for all trials. So you can see we have a participant column, a trial column. And if we scroll down, we eventually get to trial 2. This is still participant 1. If we scroll down further, we're going to get to participant 2, trial 2, etc. And so this file generally is going to have the data that you need for your analysis. And from there, the rest of the work is going to be done in the statistics program. In this case, I only included one kind of dependent measure. But I will point out, like, if you go into we have a in our data viewer frequently asked questions on the forums, there's a question, how do I extract reading measures from data viewer? And like data viewer is a general purpose tool. It's not just for reading, it's for other types of studies too. But you can see there, these are like commonly used reading measures that are just talked about and used in papers in the eye tracking and reading community. And so this is just a description, operational definitions of them. And then this describes 2 methods for getting that. There's an application called Get Reading Measures. You could try that if you wanted. But what I was just doing there was what probably will work for most cases is just making an interest area report from data viewer. And for example, if you want gaze duration, which is a commonly reported measure, it's like the sum of all fixations the first time you look at an interest area until you leave that interest area backwards or forwards, but provided you didn't skip it during first pass reading. So if you skip it and come back to it, gaze duration doesn't count, or it's 0, or however you want to treat it, basically. But the idea is like you create an interest area report and include this variable, but also include interest area first fixation was progressive, must be 1 so that it's not skipped. So you probably want to select only cases where it wasn't skipped if you want to follow like the typical operational definitions. I mean, you might have your own definitions of these variables, but this is just showing an example of how to extract these measures based on these operational definitions up there. And so like you can just kind of get all these typically reported reading measures directly from an interest area report by doing just a little bit of selection in a stats program and including the right variables for each measure. Okay. And just going back to the to this, like, let's imagine you changed your analysis goals and now you only want to focus on subtitle 3 in every video or something. Then you can use these messages to do more fine-grained interest period. So I could make a new interest period called like subtitle 3. And we can use these messages that are automatically sent out by when you're defining an interest period, you define the start up here and you define the end down here. And so for the end of the interest period, we could do subtitle offset 003. And then you would only be looking at that period for your different videos. See, you can see now we started that message and we end when it offsets which happens, in this case, happens to be the time the next subtitle onsets. But now when we made an interest area report, it would only include the data for that interest period, if that makes sense. So there are ways you can also make interest periods for particular subtitles. Okay, I think we kind of just went a little bit over the allotted time limit. So maybe this is a good time to stop. I don't know, Kurt, or...
Yeah, sounds good. Unless anyone has any questions.
Question. Yeah, question period.
Okay. Well, we had lots of questions during.
Yeah, Kurt, we'll grill you in London.
So